Alright, hey guys, what's going on? So today we are going to go back to Biophysics. This is the Above and Beyond Biophysics playlist where you're going to learn a lot about biophysics that you probably haven't learned in other places on YouTube or in any of your classes for that matter. This playlist in biophysics is going to be a very long playlist probably because I want to cover as much as I possibly can. It's a very broad subject. And as we all know, as you get more advanced into a subject, the more broad you find out that it becomes. But with that being said, make sure to hit that like and subscribe button. Make sure to go into my Patreon page if you want to support the channel. Now let's get into the content. Okay, so we are talking about proteins now. And we're going to talk in the latter part of this video just what the structure of this discussion or this course is going to be like going forward, because again, biophysics is one of those areas of physics or biology that could really go in either any direction. And so I'm going to provide you somewhat of a structured way of looking at how this course is going to unfold within the foreseeable future, not within the long foreseeable future, just because I want to focus more locally right now. And so in the last video we talked about amino acids. They were the building blocks of proteins. We're going to start on a molecular basis. We could start on a quantum basis, but I really want to get really familiar with proteins because I think if we go quantum, then that's more of a physical chemistry approach. So we're starting really with proteins. Proteins, again, as we've noted, were made up of amino acids, and these amino acids organize themselves in unique ways to make proteins. So right here we have a protein. This is specifically an ionotropic glutamate receptor, specifically a kainate receptor, where we're going to get into this stuff in large amounts of detail. But anyways, this is what a protein, this is a general protein, it's a transmembrane protein, and it has a primary structure to it. That primary structure is the amino acid sequence itself. So if you recall, we initially were talking about the DNA sequence. That's different from the amino acid sequence. So the DNA sequence, again, was the nucleic acid space. That was the space where we were talking about information, right? So nucleic acids, that was information space. And then we have this mapping to amino acid space or function space. This is a picture I want to keep in mind as we're doing this, as we're going through this lecture series, just because I think it's quite insightful. I think there's really two different types of spaces that biology encompasses. One is information, the other one is functional. And so we are talking about this dogma here of going from information space to functional space. And within functional space, we have these sequences. These sequences are made up of amino acids, right? So amino acids. And just as an example here, this is what that would look like, right? So we have an amino acid here, right? So isoleucine, leucine, and then we have glycine, and we have tryptophan, we have histidine, we have arginine, we have all of these amino acids are all linked together. They all underwent a dehydration process, right? And they can all also undergo hydration as well, where dehydration and hydration are sort of have counter effects on the DNA. As we talked about, one on one hand breaks the peptide bond, the other one forms a peptide bond, right? And so that again is the primary structure of a protein, right? It's just a sequence, nothing else. The secondary structure, however, comes in different forms. Right? So the secondary structure can come in two broad forms, one being alpha helices and the other one being beta pleated sheets, right? So here is an example alpha helix, and here's a beta pleated sheet. Beta pleated sheets are amino acids organized linearly with respect to one another, and these they undergo these interactions here, right? So these interactions are hydrogen bonds, right? So there's a lot of hydrogen bonds, and they are interacting. These nitrogens, they have a hydrogen attached to them, so that hydrogen that didn't leave during the hydration process, or that during the dehydration process, right? The hydrogen that did not leave is incorporated into the hydrogen bond here with this double bonded oxygen on the carbon, right? So these beta pleated sheets are linked together via these hydrogen bonds. Alpha helixes are similar, right? So we have 3.6 R groups, right? So these R groups, those are these guys, per turn, right? This is, it tells you approximately how many amino acids there are when you go from here, this point here, to this point here. Notice how we start at an N and we end at a C, right? So that's why we're saying 3.6, because we don't start and end at an N, at two N groups, right? The next N group, right, is like right here. So we have approximately 3.6. So we're covering 3 amino acids, right? So when we go from here and we go across here, we are passing 3 and then an extra part of a 4th amino acid as we traverse this helix. Okay? But this can actually change, right? So we have helixes called 310 helix, which is where we have 3 residues per turn. So that means that these turns are actually tighter. Okay, but then we also have pi helices, which means that we have 4.6 residues per turn. So these turns are actually looser. So we have variability in the secondary structure, right? So the secondary structure, again, branches off between beta pleated sheets and alpha helices, and then the alpha helices branch off into the pi helices and the 310 helices. Okay, so we have some variability here, and that encodes, again, the variability encodes the type of functional attributes that these proteins can have. Okay, and so this is all, again, embedded in a protein right here. So these are all atoms or amino acids, and typically actually I'll do this. Typically in a protein like this, right, we have, this is actually called the transmembrane domain. We'll go into more of this, but in transmembrane domains we typically have alpha helices. So a lot of these are alpha helices. Okay, the alpha helices go through transmembrane domains because the outlining, the alpha helices themselves, any of the R groups that are that don't like water or are hydrophobic are going to likely embed themselves in a region where you have lipids, right? Because lipids, again, are also hydrophobic. They organize themselves in a way such that all of the water sort of hating part of them is embedded in a region where there's no water. Okay, and that is this region right here. We're going to go more into this later. This is really just introductory stuff, right? Stuff that I feel like if I didn't go over, then this would make future talks probably a little bit longer. But anyways, these are the two main structures, right? And then we're going to have tertiary and quaternary structures. So if we look at this protein, so the protein has primary structure, the protein has secondary structure. Tertiary structure is going to be the overall shape of one of these guys, right? So I have this guy, say, colored in red. This guy is colored in red, and that is the tertiary structure of the protein. Each one of these is its own separate subunit. Okay? And so the quaternary structure is the structure of all the subunits coming together. Okay, so tertiary structure is the structure of one subunit. The quaternary structure is the structure of all of the necessary subunits interacting together. Okay, so we have primary, secondary, tertiary, and quaternary structure, and we have variability within each one of these structures. So variability in primary structure oftentimes will take the form of mutations. So different types of mutations include deletions. So that would be a deletion in an amino acid or a nucleic acid, leading to, again, some type of variability in the primary structure. Duplication, inversion, insertion, translocation, silence mutations, missense, nonsense mutations. These are all, again, different types of mutations that vary the degree of that vary within the primary structure, and they give different types of information depending on which one of these is occurring. This is largely the topic of genetics, but I do want to get into this a little bit, but we're not going to get into it right now. I'm really, again, just introducing us to all of this right now, and then we're going to get deeper into it later. When we talk about secondary structure, this is where things get really interesting. This is usually where protein biophysics usually kicks off. Like if you take a course, it kicks off with thermodynamics, statistical mechanics, electrodynamics, also a little bit of quantum mechanics. But the idea here is that variability in secondary structure, remember, secondary structure is the formation of beta-plated sheets and alpha helices. Secondary structure comes from protein folding, right? So the way the protein folds. And that all is determined by, again, thermodynamics, statistics, statistical mechanics, electrodynamics. This is all the physics that's involved in making proteins fold in specific ways so that they can attain some specific type of function. Then we also have types of interactions that we'll talk about. We've talked a little bit about hydrogen bonding. Cysteine bonding is also very important. I'm realizing right now I should erase this because hydrogen bonds are not covalent interactions. I'll just erase that altogether. Cysteine bonds are, though. Cysteine bonds are, and if you take a look, for example, at this amino acid sequence, we have this thing right here. This is an SH. Usually what happens in a cysteine bond is that this hydrogen is going to get kicked off, and we're going to have another S from the sequence. Another S from somewhere, right? So suppose we have, suppose this sequence contains more amino acids, and then at this amino acid, say, at this amino acid we have an S group, and a cysteine bond can form there when we dehydrate, or when we take away this hydrogen. So that's how cysteine bonds form. Cysteine bonds are very strong, okay? Usually if you want to denature a protein, you have to add something that breaks apart these cysteine bonds, and we'll talk about that as well. All right. So we have two of the most strongly holding interactions, again, are hydrogen bonding and cysteine bonding. And that's all within formation of secondary structure. Now we get to tertiary structure. So variability in tertiary structure comes in the form of different types of amino acid interactions. So we talked about hydrogen bonds and cysteine bonds, again, not entirely covalent. So cysteine bonds are covalent, hydrogen bonds are not. But we can also talk about van der Waals forces, right? So van der Waals forces they're one of the more weaker types of forces that attract molecules together, but they're still there nonetheless. The van der Waals forces, again, is the idea that surface area will play a role in this. The more surface area you have on a molecule or on a protein, the more of a chance there is to be some sort of small interactions between two molecules. And we'll talk about this as well. This, again, there's a lot of mention of this in physical chemistry. Again, I want this to be a course in biophysics, not PChem. PChem is going to be a separate playlist. And then we have dipole interactions. Dipole interactions occur when we have a hydrogen, a nitrogen, an oxygen, a fluorine, a chlorine, bromine, anything that can really form electrons, unpaired electrons, will form a dipole, right? So again, I think iodine can also be included in here, but the idea is if I have, say, some double bond oxygen, and that's attached to something here, well, we have electrons here, and so the dipole looks like this, right? So we have positively charged region right here, but a negatively charged region right here. And so if something has a positive charge, say we have nitrogen with a positive charge, like this, say, attached to something. This has a slight negative charge on this end, right? So these two guys can interact to some degree. So that's the dipole-dipole interactions. Dipole-dipole means you have a dipole here, right? So the dipole right here, and we have a dipole right here. And so the dipoles are interacting with each other. Okay. And then the hydrogen and cysteine bonds. Okay, we're pushing on 20 minutes. What I want to do here is, again, just talk about what I talked about in the initial parts of this video, which is how this discussion or how this lecture series or whatever you want to call it is going to go forward. Again, biophysics is really complex. Once you learn the initial parts, the introduction parts in biology and stuff like that, what you start to realize is that there are multiple routes you can go down, right? It just becomes, the level of complexity just exponentiates. So I'm going to add some structure to this. I'm going to tell you what the structure to this is going to look like going forward. So we're going to start with physics, right? Because I'm doing this primarily because I know there's many of you out there who are not huge fans of physics, okay? And you're more fans of biology, neuroscience, and all that kind of stuff. So I'm kind of getting this out of the way for those of you who don't really want to watch the physics, okay? I would be remiss to not talk about the physics at all. So I'm going to talk about the physics. I'm going to try to go deep into the physics, but then once that's all done and over with, then we're going to get into more protein folding, and then we're going to talk about ion channels. And this is where this channel is going to get really unique, right? We're going to talk about ion channels within the context of nerves, right? So we're really going to take an anatomy perspective on this. My emphasis, once we get to the ion channels, is going to be the physiology is very important, and how can we explain physiology largely through ion channels. Ion channels are going to help us explain how action potentials are initiated. They're going to help us understand how we are affected by drugs, for example. And depending on the drug, again, like cocaine, LSD, this is the kind of stuff you think you hear about in the news. Fentanyl, for example, this is the kind of stuff you hear about in the news, but you are not quite you don't quite understand perhaps how this all works on a molecular level, and you would like to. And so the idea here is that we're going to hit the brakes on ion channels. We're going to start talking about nerves. We're going to start talking about action potentials. We're going to start talking about concentration gradients. Okay, and then once we finish with concentration gradients, the Nernst potential and all this kind of stuff, then we're going to get into specifics. Okay, we're going to talk about the effects on different types of drugs. Okay, cocaine, again, LSD, perampanel, any type of drug that you can think of, right? I'm really intending to hit the brakes on this and really slow down to really understand and take a look at the types of channels that these drugs affect, and sort of just have that be a discussion, okay? Because I want this to be relevant within the context of your everyday life. When you go to the hospital, when you go to pharmacy, I really want this to be within that context. Then we're going to step back out of the specifics, and we're going to talk about diffusion, fluorescence, optical traps, and magnetic tweezers. Okay, and this is really going to put us into the domain of the tools that we use to study biophysics. Okay, this is going to include so not optical traps and magnetic tweezers, but we're going to go into talking about cryo-EM. So like cryo-EM, electrophysiology, separation techniques, chromatography, okay. And we're going to spend some time talking about methods, okay? So we're going from, again, thermodynamics, hitting the brakes on ion channels, and then methods. Okay, there's a whole other area of biophysics that I also want to go over, and that's going to be the math, right? So there's the physics, very simple basic physics. Physics can be explained by math, but then there's also other areas of math like stochastic processes, random walks and stuff like this that I also want to cover, and that's going to come later. Okay, but this is how a lot of this is going to go down. So with that being said, I hope you like this kind of content. Make sure to hit that like and subscribe button, and I will see you guys in the next video. Bye.